Hello, and welcome to Talking About Regeneration, the podcast that's all about stem cell and regenerative medicine and some other stuff too. I'm Kevin McCormack, Communications Director at CIRM. We're California's stem cell agency, and we have $5.5 billion to change the world. And this podcast will introduce you to some of the people who are going to help us do just that. Enjoy the show. Our guests today are two scientists who are in the forefront of trying to find new and better ways to help people with type 1 diabetes. Dr. Monsi Jamin is the Vice President for Clinical Development at ViaCyte. That's a company CIRM has been supporting for many years. Before joining ViaCyte, Dr. Jamin was the Senior Medical Director at Covance Clinical Development Services. She was also an attending physician at Harvard Medical School and Massachusetts General Hospital, where she was responsible for clinical care for type 1 and type 2 pediatric diabetes patients. We're also joined by Dr. Crystal Nitre, a co-founder of Encelin, a company that sprang out of some CIRM-funded research at the University of California, San Francisco. Both doctors are involved in developing a new way of treating type 1 diabetes that doesn't involve constant blood sugar monitoring and insulin injections. And at this point, I have to declare a bias. I find this work fascinating, and whenever I talk about it in public, people are amazed and always want to know some more. So, doctors, welcome to Talking About Regeneration. Thanks for taking time out of your busy schedules to chat with me.
Yeah, thank you so much
Thank you for having us.
That's great. So, Dr. Nitre, if I could start with you. How big of a problem is type 1 diabetes, and how does it affect people's lives?
Well, I'm sure it's no surprise to a number of people on this call that type 1 diabetes affects millions of people, over 1.5 million people in the U.S. And it is a quite burdensome disease. If you think about all of your daily activities being questioned as to, are you hungry? Do you need to give yourself more blood sugar? It affects people very, very physically and also very mentally taxing. And because of that, we got really excited about thinking about ways that we can help these patients that have such a taxing healthcare regimen that they need to adhere to.
Great. So what is the approach you're taking? I mean, first of all, what are the current methods for treating type 1 diabetes, and what are the methods you're using?
Yeah, so the standard approach for treating type 1 diabetes is insulin injection. So you deliver insulin either through an injection or through a pump based on the amount of glucose or sugar that you need to respond to. So if you are not eating, you'll deliver less insulin. If you're eating a giant meal, which I like to have giant meals, you'll have to have more insulin. And what's kind of crazy about this is also if you have a giant meal and then you're more stressed or you're less stressed or you have a good day or a bad day, that number is not always the same. So it's constantly changing. It's not necessarily sort of this very mathematical equation. And that's what makes these types of endocrine diseases so challenging, is that they are very dynamic and responding. So at Encelin, we're really excited about using the power of cells or as living medicines to be able to deliver therapeutics when you need them based on your own body's needs. And diabetes is an excellent example of that because it is a disease that has a clear mechanism of action. We know what's wrong in diabetes. We know that you need to administer insulin. The real challenge of this disease space is that you need to be able to deliver it at a specific time, at a specific dose. And cells have these innate capabilities to be able to do all of that computation and calculation for you. So we're really excited about leveraging the power of cells as living medicines to be able to deliver therapeutics when needed. And diabetes is an excellent case study for that.
Great, thank you. Monsi, and now I'm going to be switching to first names because I think it's just easier. You were a practicing physician before you came to this area. You were at one of the most prestigious medical institutions in the US, probably in the world. So what made you decide to switch to research?
So actually, I continued to see patients until end of last year. And I wanted to continue to see patients because they really informed the reason I went into research. Type 1 diabetes, type 2 diabetes runs rampant in my family. And I wanted to go into endocrinology for that reason itself. And then when I actually started seeing patients specifically with type 1 diabetes, started to see the initial shock, the burden, and then the relief that they had from just getting new therapies it was the reason that I went into research. But one thing that was really important was that I continue to see them and be able to learn from them and continue to learn from their hurdles, which informed every step of the way of my research career, from working on the artificial pancreas trials at Mass General and then moving on to biotech to really be part of a functional cure, which is what we're trying to work on at BioSite.
So talk a bit about that functional cure. What does that involve?
So, at BioSite, what we've been working on, and we've been working on this in clinic for several years now. We're the first in clinic with human data showing that cells engraft and produce insulin and glucagon. So the functional cure really is enabling patients to have cells that do what the pancreas does. So the pancreas is made up of islet cells. Islet cells, under that umbrella, is alpha cells and beta cells. Now, to simplify that, beta cells make insulin that brings your blood sugar down. Alpha cells make glucagon that brings your blood sugar up. And you really need to have both to have what the pancreas actually does for those without type 1 diabetes. A functional cure supplies these cells that enable this therapy to function as a pancreas.
How does this device know when to secrete insulin or perform any of the other functions that the pancreas would normally do?
Sure. So typically, there's a glucose response. You have glucose in the blood, and that engages the cells through a signaling pathway to either release one or the other hormone. Our cells, once implanted, actually do that. They have a signaling pathway through a response through the blood. It shows that there's blood glucose is going a certain way, trajectory up or down, and accordingly, either insulin or glucagon are released. In fact, we actually just released some data at the American Diabetes Association in June showing that our cells in fact work. And not only do they work, they bring glucose levels to the requirement that the ADA requires to have great control in patients. And it was seminal data for us, never been seen in the field. And we're really proud of that. And it's driving our company even further to make sure that we provide a cure for these patients.
That's pretty exciting. Crystal, if I could come back to you now. I read an interview where you said your job is to try and find ways to make cells happy. How do you go about making cells happy?
Yeah, it is true. I think one of the things that we're really focused on, and where I think one of the big areas of differentiation for insulin is that we're really focusing on empowering cells to do what they do best. And with that, our preclinical research has been really focused around helping cells to be able to survive in the body. We want to be able to both protect these cells so they can provide these very sophisticated, timely, and dose appropriate responses. And at the same time, also protecting the patient so that we can have these cells be safely put in the body. And at Insulin, I think that those are really the big exciting part, is having this nice encapsulation approach that allows you to take cells from a wide variety of sources, let them do their thing, help them be happy. And it's true. I do think a lot about helping cells be happy in the body because, they have to survive to be able to be functional. They have to be happy. They have to have all of those right environmental cues to be thriving and successfully therapeutic. And that's really, going back to being happy, that's really what we're focusing on insulin, is helping these cells survive to be able to provide valuable therapeutic output.
Well, that makes sense. I think we all do a much better job when we're happy with our work. I know I do. But the body typically doesn't like having things, objects, anything really, shoved inside it, and often mounts an attack against that object. So how do you protect your device from the body and the body from your device if things go wrong?
And thank you so much for asking that question, because I think this is an area that we're very excited about at Insulin, is to be able to not only protect these cells in an immunological sense, as in you can take a foreign tissue and put it in the body and help these cells maintain viability. For example, Kevin, if I were to take your cells and put them in myself, my body would reject them because they're not crystal cells. Exactly. But if I were to take my own cells, put them back, and then they'd be fine. So we've shown in a number of animal models that we can successfully do this. Obviously not, I can't take Kevin's cells, but we can take mouse Kevin cells, put them and so forth, and do those types of transplants and show that they're protected. And beyond the ability to protect these cells and Insulin's technology, we've also shown that we can take these, foreign encapsulation products, put them in animals, and show that they're also not seen as foreign bodies and they're not reacted with a foreign body response. We're getting a really beautiful level of engraftment that shows that these cells can be happy. Again, thinking about helping cells be happy is really core to what Enslin's product is focused on, is keeping those cells protected, successful, and well engrafted in the body.
Monsi, looking at the device that ViaCyte has developed, it's quite small, about half the size of a business card. So what's the advantage of having an implantable element like that?
Well, there's several benefits here. So it's an implantable device. The cells are encapsulated. They are not released outside of that device. I think some of the points that you and Crystal hit on is how do you make the cells happy, and how do you make them last, and how do you make them viable and effective? And so the benefit of an encapsulated product, and specifically, and I'll go back to kind of the data that we presented in July, it was from a product that actually has direct vascularization into the device, which means that blood vessels go in, they supply nutrients, they allow the cells to survive, they allow the cleaning cells of the body to go in and clean out the cells and continue to do the mechanisms that are required for cell viability overall. So I think part of encapsulation needs to include a way for cells to survive long term. And I think that's one of the bigger pieces, and it has to be sustained. The nutrients have to be able to get in, and insulin glucagon have to be able to get out. So those are the things that we've worked on over the years to really finesse and improve and optimize.
How long theoretically could one of these devices work and last? I mean, would you have to take them out on, say, a yearly basis and replenish the cells?
So, what our proof of concept of the work that we've done has been really informed by islet cell transplants. And, those are injected cells, and often they lasted average approximately 5 years. Now, with encapsulated product, we hope that we can reach that. Right now, our longest study has been out to 2 years, and we've been able to maintain those devices in patients for that long. And as we grow and improve and develop our studies, we hope to find that answer. I think what's viable is 3 to 5 years for these types of encapsulated methods.
Crystal, I know that ViaCyte is already in clinical trials. When do you anticipate that Ensilin will be able to test its device in people?
One of the things that to move into the clinic, we're really focused on two things. One, keeping the product safe for the patient. And the second one is these patients are really waiting for us. And I think one of the great things that was mentioned earlier is that this is a high unmet clinical need. And they're waiting for a therapy. I think these patients are high risk. So one of the things that we're thinking about to move quickly into the clinic is getting there quickly and safely. We're looking to be in the clinic as quickly as we can to get there, but we have not yet released a date out into the world. But we're working on it quickly, and we're really excited about our progress.
One of the problems when you talk about projects like this is that it inevitably leads people to ask, when will this be ready? When can I get one? Monsi, what do you tell people when they ask you that question?
At this point, what we're focused on is ensuring that we have more data. We're on track to provide additional patient data, especially from our PEC Direct Phase 2 product candidate in the first half of next year. We're also really working with our world-class leader, CRISPR Therapeutics, for immune evasion, which is a really exciting next step, I think, in stem cell research, is making the cells hide from the immune response in the body. And we're starting that clinical trial at the end of this year, and hoping to have more data. And then that's really going to inform when we're going to be ready for patients. So I think we're close. We're closer than we've ever been.
This is really amazing stuff, and I want to thank you both for being here today, Dr. Monsi Jamin and Dr. Crystal Naitre. Thanks for coming in and chatting to me, and good luck with your research. I look forward to sharing stories of your success in future podcasts.
Thank you so much
for having us.
And thank you at home, in the car, the shower, or wherever you listen to podcasts. I'm Kevin McCormack, and I'll see you next time on Talking About Regeneration.